In this video, we'll talk about the small intestine and the large intestine. So switching over to a picture, we can see the intestines here. We know that when the acidic chyme leaves the stomach and moves into the small intestine, that it's going to be processed by secretions from the pancreas, the liver, and the gallbladder. We talked about that in the last video. And so now that we know what processing is going on within the small intestine, let's first take a look at the regions of the small intestine and talk about the work that the small intestine does. So to zero in on that a little bit, here's a color-coded picture of the small intestine. It's regionalized. We can see the three regions. The first one is the duodenum. We've talked about that one already. It's highlighted blue here. And of course, this is where the pyloric sphincter would be, and the stomach is removed in this picture. Of course, the pancreas is tucked away in here, and this is where that hepatopancreatic sphincter and the secretions from the liver, gallbladder, and pancreas enter in. And that's all removed just so we can see the small intestine. So that first region, the duodenum, is the shortest region of the small intestine. It's also the most stable. It's pretty fixed in place because the head of the pancreas is nested right in there, and then the stomach is attached here. The duodenum leads into a second region called the jejunum. One difference between the two regions is that the duodenum has a lot of mucus glands. They're called Brunner's glands, which makes sense because that mucus is going to protect the the duodenum, that initial portion of the small intestine, from the acidic chyme that's moving into this region. The jejunum, it's longer, it's more movable, and the jejunum leads into the third region, the distal-most region, the last one, called the ileum. And the ileum will attach to the large intestine, which we'll take a look at. So the ileum is the longest region. In the submucosa of the ileum, there are a lot of lymph nodes. That's a distinguishing factor of the ileum, which also makes sense because we're absorbing nutrients, right? And so when we ingest food and fluid, we're ingesting nutrients, but I bet we're also ingesting some other things that we don't want. You know, like maybe pathogens, parasites, right? Who knows? So things that we don't want, and it makes sense to have a whole army of lymph nodes here in the ileum so that when things are absorbed that shouldn't be there, we can attack that. So that gives you a little overview of the regions. Just looking through my pictures here. In this picture, what they're doing is they're lifting up the greater omentum, and here we can see the small intestine. And you see what's attached to this connective tissue sheet? That's an extension of the visceral peritoneum. That's what's known as mesentery. It's kind of cool to see in real life if you ever dissect a large organism, cat, fetal pig, something like that. It's the same as humans, that you can unravel the small intestine and see the mesentery and see the rich blood supply actually branching through the mesentery. So let's leave the small intestine in general and take a closer look at the wall of the small intestine. So here is a picture that is kind of zeroed in on the mucosa. And actually, before we get that close, let's take a look at this picture. Okay, so in this picture we can see that the mucosa of the small intestine, it's similar to what we saw with the stomach. So in the stomach, we saw that it wasn't, smooth, that the mucosa was thrown into these folds called rugae. And we can see the same thing here. These folds are called plica, or plica circularis. And what they do is it's kind of like a pleat of mucosa that juts into the lumen. And what it does is it greatly increases the surface area. Not only that, but take a look at the surface of the mucosa. Do you see how it's covered in all those finger-like extensions? Those are called villi. And so the villi also are going to, increase the surface area through which we can absorb nutrients. So the primary function of the small intestine is absorption. I know we've talked about that a few videos back. Keep in mind, absorption is really critical. It's really kind of the key function of the digestive system, because unless we can move nutrients from the lumen into the blood, then we really haven't done much, right? So the villi will help. And on top of the villi, let's take another look at this. So here you can see the plica, you know, those pleats. We can see the villi, those little finger-like extensions on the cell surface. Here's a cadaver shot. This is what the plica look like inside of the small intestine. So it's really significant, maybe more than, you know, you would expect to see, you know, just with your bare eyes. And so taking a look at this picture of the villi, do you see what they're doing here is they're zeroing in on the surface and showing us that there are basically villi on the villi. And so do you see all these little extensions that they're drawing? Those are what you call microvilli. Microvilli are just finger-like extensions of the cytoplasm. They're not like cilia. They're not motile. They're not going to move anything. Again, they're just increasing the surface area through which we can absorb nutrients. And so, you know, this is a crazy example of increasing surface area because we have the plica circularis, we have the villi, and now we're seeing that on top of the villi we have the microvilli, okay, which I'm kind of like sketching in there. So I hope that makes sense. Now, taking a closer look at the mucosa, you know, in addition to the villi, to zero in on that a little bit, notice how there are pores in between the villi. Those are intestinal glands. They're also known as the crypts of Lieberkühn. So either way, they're intestinal glands. And so this can be tricky when you look at the slide until you get used to this, because this is a villus, right? And then this part is that invagination from the surface. That's an intestinal gland. So the villus, the epithelial tissue here, is still simple columnar. It's simple columnar from the stomach right through the end of the large intestine. So we see the simple columnar. You probably recognize the goblet cells within the simple columnar. We know that the connective tissue right below the epithelium, this loose connective tissue, it's nicknamed the lamina propria. We can see the capillary beds in the lamina propria, and you can see the lymphatic capillary in the lamina propria. And one thing to notice here is that each villus, each of those finger-like extensions, will have its own capillary bed and its own lymphatic capillary. We can see mucus glands, we can see lymph nodes. Here's the muscularis mucosae, so that's the boundary of the mucosa. And then this would be submucosa, where we can see some, you know, larger glands and some lymph nodes. So I hope that makes sense. I'm going to take a look at another picture. So this is from our book, and here they're just zeroed in on one villus. So what we want to notice here is that the villus, just what we already noticed, the simple columnar with the goblet cells, the microvilli on the villus, that the capillary bed, the lymphatic capillary, the nerve supply, and so each villus has that, which makes sense because if you have a nutrient out here in the lumen, that nutrient is going to have to be absorbed through that simple columnar, and the nutrient is either going to enter into the blood, or we're going to see today how if that nutrient is fat, that the fat is actually going to get absorbed into the lacteal. Okay, so sugar and protein get absorbed into the blood, fats get absorbed into the lacteal. I'll just set the stage with that, so we'll talk a lot more about that in this video. And just looking at a bigger picture of all of the layers of the small intestine, we can see the villi, we can see the intestinal glands in between the villi, the lamina propria, the muscularis mucosae, the submucosa, the circular coat of smooth muscle, muscularis externa, the longitudinal coat, and then the serosa. So the main modification that we're seeing in the small intestine is this great increase of surface area with the plica, the villi, and the microvilli. So looking back at the notes, I try to make a note of these things. Let's see. So the regions are the duodenum. Okay, that's that proximal C shape. That's where we have the Brunner's glands that secrete mucus that protect the duodenum. Next is the jejunum, which is more movable. It's attached by mesentery. Okay, bulk of digestion and absorption occur there. And then the distalmost portion is the ileum. And this is where we can see all of those clusters of lymph nodes in the lamina propria. Those are known as Peyer's patches. A Peyer's patch would just be a little collection of lymph nodes within the lamina propria. So as far as structural modifications, we saw that that's mainly to increase the surface area, and those include the plica circularis, the villi, and the microvilli. So we looked at all of those. So what I do in the notes, other than going through the anatomy, is to talk about the function. I really just go through those 4 functions that we looked at on page 1 and page 2 of the notes. What is the small intestine doing as far as secretion, digestion, absorption, motility? What is the role of the small intestine? And just to look at this quickly, so you can see I'm talking about secretion, and here I'm talking about digestion, chemical digestion, the breakdown of macronutrients into micronutrients. Absorption is the main job. And so you can see I have a lot of notes about absorption, some pictures for us, a lot of pictures about absorption. And then down here at the bottom of page 15, I'm talking about motility. And so I just wanted to kind of show you the overview. So all of those pictures and notes just have to do with the absorption that occurs within the small intestine. So starting at the top, the secretion. There is secretion by the small intestine. One thing that we already know is the mucus secretion. Mucus would come from the Brunner's glands and also from the goblet cells. And then remember the intestinal glands that we saw in between the villi? Those intestinal glands secrete watery fluid. And what that watery fluid does is it facilitates absorption. So the small intestine does not secrete digestive enzymes like the pancreas does. The secretion coming from those glands is really kind of watery. And I guess I would describe it like this, if this makes sense. If you ever had, say, like a powdered drink mix in water, and you drink it like emergency, something like that, and there's still like powdery residue, maybe you'd add more water and swirl it around and, try to drink the rest of that fluid. That's how I think of this intestinal secretion. It's just a watery secretion that can mix with the nutrients to solubilize the nutrients and make them easier to absorb. That's all. So digestion. Digestion does occur in the small intestine, and let's just remember what we have talked about already. In the small intestine, this is where the action of the pancreatic amylase, pancreatic lipase, and the 3 pancreatic protein digesting enzymes, trypsin, chymotrypsin, and carboxypeptidase, those are all having an effect in the small intestine. But as far as the small intestine itself, it doesn't technically secrete digestive enzymes. What it does is it has digestive enzymes embedded in the plasma membranes of the simple columnar epithelial cell. And it has enzymes that will, just like the pancreas, digest every category of nutrient. Those enzymes are called brush border enzymes. And I made a table to show you those. So the proteins get digested by enzymes that are known as proteases. These proteases are going to take the peptides that were elicited by pepsin, trypsin, etc., take those peptides and chop them into individual amino acids. Those individual amino acids can be absorbed into the blood. The fat is digested by lipase, and lipase, this is just an addition to the pancreatic lipase. There's lipase embedded in the wall of the small intestine. And that lipase will elicit like single fatty acids and monoglycerides. We'll take a look at a picture of this in a little bit, so I'll be able to show you that. And then carbohydrates. There are many different categories of carbohydrates. These are disaccharides, sucrose, fructose, lactose. Actually, I'm saying the sugar names. The enzyme names have the ACE. So sucrase, fructase, lactase. Those are all enzymes that will break the disaccharide into the monosaccharide. So in other words, each of the disaccharides have their own enzymes. You don't have to worry about memorizing the names of those. But that's how that works. That's how we're going to break the disaccharides into monosaccharides. and that is digestion. So as far as absorption goes, that's the major function, like we said, of the whole system, I would say, and definitely of the small intestine. So 9 liters of food and secretions enter the small intestine per day on average, but only half a liter will exit. So that speaks to the amount of absorption that is occurring in the small intestine. So really what we care about is we care about how are the nutrients being absorbed. And so I gave you a little preview in the video. I hope you remember, because this part is easy. Sugars, okay, the monosaccharides, and amino acids. So the building blocks of the sugar, building blocks of proteins, monosaccharides and amino acids are absorbed in the same way. What happens is we use active transport to get those nutrients into that simple columnar layer, and then we eject those nutrients into the interstitial fluid by facilitated diffusion, and then they get picked up by the blood. And that's how we absorb sugar and protein. Taking a look at this picture, this is a picture from a different textbook. It's a picture from the Saladin textbook, which is an amazing textbook. We're not using it though. This is ours is good too, but this picture I thought would be really helpful. So here we're looking at sugars, proteins, and fats, how they're being broken down in the mouth, stomach, small intestine, and here's the semicolumnar of the small intestine. So with carbohydrates, we know salivary amylase is going to break down carbohydrates starting right away in the mouth from polysaccharides into disaccharides. And the next time we really work on these is in the lumen of the small intestine with pancreatic amylase. And then these disaccharides, in other words, these dimers, these 2 monosaccharides linked together, will get broken down by these brush border enzymes. So they're giving the names of the brush border enzymes. Once they're broken down, they're actively absorbed, for the most part, in the simple columnar cells, ejected, and then they get by facilitated diffusion and then picked up by the blood. For protein, we know proteins are big chains of amino acids. Nothing happens in the mouth, but in the stomach, pepsin is going to start to chop those apart into small peptides. And then in the small intestine, the trypsin, chymotrypsin, carboxypeptidase is really going to start to work on those small peptides, giving us, smaller pieces, some dipeptides, and then we have peptidases that will break those down into individual amino acids that will be actively transported into the simple columnar. Move that up a little bit, and then diffuse out and then enter into the blood, and they're absorbed into the blood. And so the main thing that I want to look at in this picture, because this picture is kind of small, is that fats are different. Like where we're getting really the significant fat digestion the first time is pancreatic lipase. So there is lingual lipase, gastric lipase. We didn't cover these in the class. The significant breakdown is the pancreatic lipase. And all I want to show you in this picture is how when fats are absorbed by the small intestine, that they're not entering into the blood. As I mentioned earlier in the video, they're going to enter into a lymphatic capillary called the lacteal. So that's a big difference. I go through all of the steps on how that works, but I'm just going to look at some pictures to describe it, and then we'll go back to that text and make sure it makes sense. So this is a picture from our book, and I just thought that you could mark this up if you wanted to. So what they're showing here, and I just wanted to remember that in the lumen of the small intestine, because it's watery out here, remember that the secretion of the small intestine, it's mainly water, coming from the intestinal glands. What happens to fat in water? We know it's going to happen. It's going to aggregate into this one big globule because it's hydrophobic and it doesn't interact with water. And so the only reason that we're seeing this solubilization of fat, and I drew this in myself, so this part isn't in the book, is because bile salts are emulsifying the fat. So when you go from one big fat drop to many fat drops, that's known as emulsification. And this is the job of the bile salts. So the bile salts are emulsifying the fat, and this is going to allow the pancreatic lipase to get in there and to digest the fats. So this structure is known as a micelle. A micelle is all of this fat that's, kind of aggregating together, and it's, solubilized in the watery solution by the bile salt. So inside of the micelle would be the fatty acids, the monoglycerides, cholesterol, things like that. So in order for fats to be absorbed, what I'm trying to show in my picture is that the bile salts get absorbed separately. So the bile salts will get absorbed, and when the bile salts get absorbed, what they do is they're going to get absorbed into the blood, and we know that the blood is going to the liver, right? That's the hepatic portal system. It's going to go into a hepatic portal vein, go to the liver. Perfect, because the liver makes bile, and so what the liver will do is remove these bile salts from the blood and recycle them and use them again, put them back out in the bile to do this job again. The fats need the bile salts to be solubilized in this watery environment. The fats don't need the bile salts to be absorbed into the cell, right? Because this is a phospholipid bilayer. And so just like the hydrophobic steroid hormones that we talked about, this fat, it's hydrophobic, that's going to just be able to diffuse right into the cell. And when it diffuses into the cell, it is processed by the cell. The cell actually remanufactures triglycerides. So it will take the individual fatty acids that cross the membrane, the smaller fats, manufacture triglycerides. And then what the cell does is it puts a protein coat on that fat, and that is known as a chylomicron. The chylomicrons, in other words, the protein-coated fat, let's call it, get packaged in membranous vesicles by the Golgi, and then those membranous vesicles are going to release the chylomicrons by exocytosis into the interstitial fluid. Now, the chylomicrons are going to be absorbed by the lacteal. Okay, so this is the lacteal. This is not a blood vessel. So in other words, the fats will be absorbed by the lymphatic system. And the reason for that is, kind of what we were talking about over here. You know how all of this blood is heading over to the liver through the hepatic portal system? And so the liver is going to process that blood. It's going to metabolize the sugar, the protein that's in the blood. And the liver will also process and metabolize fat. But the liver doesn't want to be bombarded with 100% of our dietary fat. It's not good for the liver. And so instead of the fat being absorbed into the blood and then hitting the liver 100% of our dietary intake of fat, it gets absorbed in the lacteal. And hopefully you remember this from earlier in the course. We know that the lacteal is going to be part of the lymphatic system, right? So the lymph circulates through lymph nodes, it gets cleaned up, and then ultimately the lymph empties back into our blood through the subclavian veins. So that's where this fat is going to ultimately enter the blood, through the subclavian veins as part of the lymph. Once it enters the blood, then it's evenly distributed to all of the organs, including the liver, and the liver will process that fat. So I hope you can see, by the fat being absorbed by the lymphatic system rather than the blood, it avoids the liver having to process, 100% of the fat. It just gets like a proportional amount of the fat with all of the body tissues. So this picture from Saladin, I thought, was a really nice picture. Let's look at this, and then we'll take a look at the notes. So first is emulsification. When this fat makes it into the small intestine, it's going to be in this big globule because it's in a watery environment. And so it's the bile salts that emulsify the fat. Here, they're calling this a bile acid. A bile acid is the same thing as the bile salt. Okay, so I don't mean anything different. So bile salts are solubilizing the fat, and that is going to allow the pancreatic lipase. It's going to give the surface area for the pancreatic lipase to get in there and digest the fat. And here we're looking at a triglyceride. So remember, a triglyceride is a glycerol molecule with these are the zigzag lines, 3 fatty acids coming off of it. So pancreatic lipase, what it does, it chops off, you can see those 2 peripheral fatty acids. So we get 2 free fatty acids. The one fatty acid is still bound to glycerol. We call that a monoglyceride. This is the end result of lipase digestion. These items can diffuse in to the simple columnar cell of the small intestine. And so also, just to look at this. You know, not only will it be the fatty acids and the monoglycerides, but cholesterol, fat-soluble vitamins. So all of the fat within our diet will be part of these micelles. They're going to be part of the fat droplets that are solubilized by the bile salts. And so in order for the fats to be absorbed, these micelles will just separate. The fats diffuse in, the bile salts diffuse in separately. The fatty acids, monoglycerides, actually get reprocessed into triglyceride. And then you can see a protein shell gets put on this fat, and that's known as a chylomicron. So the chylomicron is made by the simple columnar cell. It's basically protein-covered fat. I mean, that's very clever. Think about that. If you don't want fat to enter into the blood, remember how when proteins slipped out of the blood, those proteins will return to the blood through the lymph because the proteins are too big to enter into the capillary? Well, look at this. We're putting protein around the fat. Okay, we're going to make it impossible for the fat to go into the blood. We're going to make it too big. And then those chylomicrons get packaged into membranous vesicles, ejected into the interstitial fluid, and then taken up by the lymphatic capillary. So I hope that makes sense. I did write this out. So here are the steps. See if this makes sense. So fats are not going to be absorbed by the blood. Instead, they're going to be absorbed by the lacteal, and that's in order to avoid sending 100% of the ingested fat, you know, in one event to the liver. So first, the bile salts emulsify the fats in the small intestine, and then they're dispersed in the small intestine as micelles. Pancreatic lipases will digest the fat within the micelles, and then the digested monoglycerides, fatty acids, are going to just diffuse into the cells of the small intestine. Those absorbed fats are converted back to triglycerides by the smooth endoplasmic reticulum, and then these fats get covered by a protein coating, and we call that a chylomicron. The Golgi is going to package chylomicrons into a membranous vesicle, so this is just a means to eject this from the cell, right? And then using exocytosis, it's going to get those chylomicrons out of the cell into the interstitial fluid. They're going to be too big to enter the capillary. And so what happens? We know this at the end of AP2. Lymphatic system takes them in. So the chylomicrons are going to enter the blood ultimately, eventually, right through the thoracic duct and also the right lymphatic duct. It's just that the left duct, the thoracic duct, is larger. That's why I'm listing that, but it would also happen on the right. And so to wrap up the small intestine, motility is definitely a function. We see two types of motility, what's called segmenting contraction, which is kind of like moving back and forth, and then slowly moving forward. And then when nutrient absorption is complete, peristalsis, which we know, is just going to move the contents through the small intestine to the large intestine, and at the end of the small intestine there is a valve there. It's called the ileocecal valve, and that's where the ileum connects to the first ileum, the last portion of the small intestine, connects to the cecum, the first portion of the large intestine. And so we'll move on to the large intestine. And now on to the large intestine. So the small intestine is continuous with the large intestine. Let's take a look at a picture. So here we can see this is the ileum. Okay, so they removed the small intestine just so we can see the portions of the large intestine better. Here's the ileum, and then this area where the large intestine starts, this kind of like dead end, let's call it. People describe it as a blind pouch. That's what's known as the cecum. There is a valve in between the ileum, the distalmost portion of the small intestine, and the cecum, that initial segment of the large intestine, and it's called the ileocecal valve. Good name. When a peristaltic wave pushes contents through the small intestine in this area of the ileum, this valve opens and it allows the contents to move from the small intestine into the large intestine. And contents stay in the large intestine for much longer than the small intestine, which we'll talk about. But just taking a look at the anatomy of the large intestine, that's what I have in the notes to start. The cecum we've seen. Okay, so that's kind of that dead end. And then there's a little finger-like extension that comes off of the cecum. That's the appendix. And we've all heard about the appendix and problems with the appendix. As you can see, we need to keep the contents in the large intestine moving through the large intestine, which means the contents have to travel up, and then they have to travel over to the other side of the body, and then they travel down and out of the body. And so it's possible that some residue could be caught in the appendix, and that would be a perfect setup for inflammation, infection, that kind of thing. And that's why, you know, the appendix can give us a hard time sometimes. So anyway, that's the appendix. It comes off of the cecum, and this region, major region of the large intestine, is called the ascending colon. And I'll mention that that term colon and also the term bowel are equivalent to large intestine. They're synonyms. So you could say large intestine, bowel, colon, same thing. Okay, so ascending colon travels up the right side of the body, and then you can see approximately like, you know, a 90-degree angle kind of thing into the transverse colon. So that is called the right hepatic flexure because it's on the right side of the body, also known as the hepatic flexure because the liver is on this side. The transverse colon travels from the right to the left. They're showing us like a nice, you know, straight region there, but many times, you know, it's not quite so straight. If you look at medical imaging, it varies, you know, a little bit, like from individual to individual. And then on the left-hand side, there's the left colic flexure, which is just this bend, also known as the splenic flexure, again, because the spleen is on the left, if that makes sense. So either one of those names is good. Descending colon travels down, and it will start to turn a little bit like a backwards S. That's what's known as the sigmoid colon. The rectum is about the last 15 centimeters of the large intestine. And then where the rectum starts to move through the pelvic floor, that's what the part of it that we call the anal canal, which leads to the anus, which is just the opening in the large intestine. So hopefully those regions make sense. If I jump back over to the notes, basically I just listed these. So let's see, we looked at these: the cecum, the appendix, the ascending colon, moving into the hepatic flexure, transverse colon, splenic flexure to the descending colon, sigmoid. Yep, okay, so the anal sphincters, I'll show you those in a few minutes. Okay, so we'll put those on hold. So as far as the wall of the large intestine, it's pretty straightforward, but there is a little specialization that I'd like to show you. So let's take a look at a picture. So you look at this slide in lab, and I would say the one thing to notice about the large intestine is, do you see how smooth the surface is? And so that's characteristic of the large intestine. It's also characteristic of the esophagus. But the esophagus, of course, has the stratified squamous out here. Here we can definitely see the simple columnar with goblet cells. And as a matter of fact, I think that we usually use this slide in AP1 as an example of simple columnar with goblet cells. So what we're seeing here, even though that has that like nice smooth surface, is you're seeing these pits move in. These are intestinal glands. Okay, they're exocrine glands. Like, this is a really good depiction of one. So that area that I'm showing, this is where the mucus and the watery secretion would move through that and right onto the surface. So the large intestine is, you know, it has that smooth surface. So it's not like thrown into the folds like the rugae, or you don't see the villi like the small intestine. So that's a little bit of a hint that it's the large intestine. So it's simple columnar with goblet cells. Or to take a look at all of the layers, just a diagram, we can see when we look into like this picture how the folds of the mucosa are missing, and that's what we're noticing at a microscopic level. So there's no rugae, there's no plica, you know, nothing like that. It's just pretty smooth, but we do have all of these pores. This reminds me of the stomach a little bit, all of those pits, and those lead into, we just call these intestinal glands. And those intestinal glands, I'll increase the size of this, are lined with simple columnar with goblet cells. Underneath the simple columnar is the lamina propria, with, we can see the capillary bed being drawn in there. Here's the muscularis mucosae, so that's the boundary of the mucosa. Here is the submucosa, We can see the blood supply going through the submucosa, that little yellow stuff they're drawing in there, that's the nerve supply. And here we can see the muscularis externa. So here's the specialization that I wanted to tell you about. So I see that inner circular coat. That's this part of it. But then the longitudinal coat, notice how they're not drawing muscle here and they're not drawing muscle here. And that's because the longitudinal layer, the outer layer of the muscularis externa, is very minimalized in the large intestine into 3 bands. And these bands are called the tinea coli. Okay, so that's tinea coli. And we'll look at that from a bigger picture. And then the visceral peritoneum would be the connective tissue coat, or we would just say serosa. So taking a look at another picture. Let's take a look at this picture. This is from our book. Move this back down. So when we take a look at a picture or a model of the large intestine, notice this, it looks like a gray, ribbon. That's the tinea coli. So the tinea coli, that's just a longitudinal band, and usually there's 3 bands of longitudinal muscle. You can actually see it on the outside of the large intestine. So we don't have the whole, the longitudinal layer is not going to encircle the whole thing. It's reduced to these 3 kind of like ribbon-like pieces. And because of the tension in the longitudinal layers, it creates these pouches that we're seeing. So this is what I'm talking about. You see how it's kind of like bulging along the length? Those are called haustra. So haustra are created by the tinea coli. If you ever do a cadaver dissection, you'll notice that the large intestine doesn't look like it has haustra, and that's because of the laxity. After this muscle stops contracting, those little pouches go away. So those pouches are just created by, like I said, the tension in that muscle. And so the gross anatomy and microscopic anatomy, pretty straightforward. And actually the large intestine is not a vital organ. We can live without the large intestine, although it's much, more convenient and pleasant to live with the large intestine, because what it does is it receives the contents. So it will receive contents from the small intestine. Remember, absorption largely occurred in the small intestine. So when you look at, what is the function of the large intestine? To move through the large intestine, all that really is going to happen here are the contents that are there just keep getting wringed out. We remove the water from these contents as they slowly move through. So we take out as much water. So water absorption occurs. And that's how we create feces. Okay, so this is fecal matter, the material that moves into the large intestine. And so it makes sense that the shorter amount of time that this material stays in the large intestine, the more watery it is, like diarrhea, or the longer it stays in the large intestine, the more solidified it becomes, like constipation. And so, one of the biggest things we're doing is really just extracting more water. We also do extract more sodium, or I should say absorb. Okay, so we're going to absorb water and sodium from these contents, but really the major absorption has occurred in the small intestine. This is indigestible material for us, but the colon is colonized by bacteria. I'm sure you've heard of the microbiome, the gut microbiome and probiotics. So there's over 800 species of bacteria that live in our colon. Okay, they gain entry easily from the outside of the body, and they have a symbiotic relationship with us. They don't harm us, and they actually help us, because some of this material that we can't digest, the bacteria is able to digest. And when bacteria digest the indigestible fiber and sugars, it can many times extract more energy and more and produce vitamins. And so that's some of the benefit we can get from bacteria. Bacteria also create gas as a byproduct, and that's where intestinal gas comes from, some of it. Most intestinal gas, and believe it or not, our colons have liters of gas in them. On average, 7 to 9 liters of gas are contained within the large intestine, and about a half a liter a day is ejected from the large intestine. And most of that gas is swallowed air, but some of it is gas that's produced as a waste product by these bacteria. So let's jump back over to the notes. So the tinea coli, that's what you call the longitudinal layer of the muscle, just because it's not a full circumference of the longitudinal layer. it's reduced to 3 strips, each strip being known as the Tinea coli. This is singular. And then the haustra are the pouch-like structures that we see along the length of the large intestine. And so I have these notes organized by function, as we've talked about. So as far as secretion goes, we know that there are intestinal glands, and really all they do is they secrete, a salt water type fluid, so very much like the small intestine. We also have goblet cells, and we know that the goblet cells secrete mucus. As far as digestion, remember, digestion has really largely occurred in the small intestine. There are no digestive enzymes that are secreted by the large intestine. The only digestion here is going to occur by the bacteria trying to digest what we were unable to digest, aka fiber, as one word. And sometimes they can produce vitamins that we benefit from, and they produce intestinal gas. So as far as absorption, if there are any nutrients that are elicited by the bacteria, we would absorb those. And then otherwise we're just, like I said, wringing out the water, getting back some water and salt. Motility. The large intestine is a very lazy organ in that usually like what's called a propulsive mass movement will propel contents of the large intestine forward in the large intestine about 2 to 3 times per day. And so I'm going to switch over to a picture to show you that what I'm talking about, or we'll look at a different picture, is, same thing though, is that a propulsive mass movement would just say take some contents, some fecal matter, and move it in the right direction. Just in the transverse colon, as an example, contents usually stay 3 to 4 days just in the transverse colon, and so it's possible that this is a very slow journey many times. Let me show you some other pictures that I have. Here's the ileocecal valve. This is a cadaver shot. So here we're opening up the cecum. We can see the appendix, and we can see where the small intestine is leading into the cecum. But here's what I wanted to show you. This is the distal end of the rectum. So this is what we call the anal canal. And so remember how we have the circular coat and the longitudinal coat of muscle making up the muscularis externa? That circular coat is what creates the internal anal sphincter. So the internal anal sphincter is just kind of like a specialized, the distal circular end surrounding the distal portion of the anal canal. That's smooth muscle. Okay, it's part of the muscularis externa. And then within the pelvic floor, we have the external anal sphincter. That's skeletal muscle. And so the way that defecation works is very similar to urination in that if a propulsive mass movement pushes contents into the rectum, we would feel that stretch. It would go into the spinal cord, and we would sense that as the need to defecate, which, because the external anal sphincter is skeletal muscle, we can consciously keep this contracted if the time is not right. And so when we consciously decide to relax the external anal sphincter, it will elicit a reflex response, a defecation reflex. So when we relax the external anal sphincter, the internal anal sphincter will also relax, and the rectum contracts, and that would eject fecal matter. Like I said, that's known as defecation. So, not sure how you feel about ending a year-long course with this topic, but that'll be it. Thank you.